Shrushti Joshi, a student in Pune University from Department of Biotechnology. Currently, I am pursuing my PhD degree. Today, I'll be presenting on fundamentals of genome scale metabolic modeling. Along with that, I'll be talking about some basic commands that we use in Cobra Toolbox, GAPSEC, and Linux. Mentions genome scale metabolic modeling. These three computational tools pop out, that is Cobra Toolbox, GAPSEC, and Linux. Today I'll be talking about them and the commands we use for modeling basis. Along with that, I'll be giving a brief glimpse into the history and fundamentals of GEM, which is short form for genome scale metabolic modeling. A GEM computationally describes a whole set of psychometry-based, mass balanced metabolic reactions in an organism using gene-protein reactions, which are also called as GPR associations, that are formulated on the basis of genome annotations data and experimentally obtained information. The prediction of metabolic flux values for an entire set of metabolic reactions using optimization techniques such as flux balance analysis (FBA), which uses linear programming. The first genome-scale metabolic model was done for Haemophilus influenza RD and was reported by Edwards and Paslon in 1999. As of February 2019, we have almost 6,239 organisms which are either manually reconstructed or by using automatic GEM reconstruction tools. A total of 183 organisms out of the 6,239 was being subjected to manual GEM reconstruction. The GEMs for model organisms that have high scientific, industrial, and/or medicinal values have been updated several times since their initial reconstruction, as more relevant biology information keeps on popping up over the years. Currently, we have E. coli, B. subtilis, M. tuberculosis, Methanecercina, acetovirens, S. cerevisiae, C. rehin-hardy, C. elegans, Arabidopsis thaliana, and Homo sapiens. A table can be seen to your left. The applications of GEM are production of chemical and materials, drug targeting in pathogens, prediction of enzyme functions, pan-reactome analysis, modeling interaction among multiple cells or organisms, and many more. Or COBRA Toolbox. The word COBRA stands for the Constraint-Based Reconstruction and Analysis Toolbox. This is a MATLAB software suite for quantitative prediction of cellular and metacellular biochemical networks with constraint-based modeling. It implements a comprehensive collection of basic and advanced modeling methods. This includes reconstruction and modeling generation as well as biased and unbiased model-driven analysis methods. It has found widespread application in biology, biomedicine, biotechnology, and various other fields because its functions can be flexibly combined to implement tailored COBRA protocols for any biochemical networks. We use this software for modeling, analysis, and predicting a variety of metabolic phenotypes using genome-scale biochemical networks. The COBRA file is a package for constraint-based modeling of biological networks written in Python that provides a simple interface to metabolic constraint-based reconstruction and analysis. The package includes simple object-oriented interfaces for model construction and implements commonly used COBRA methods such as flux balance analysis, which is very important for GEM analysis, flux variability analysis, and gene deletion analysis. We can see there is an installation flowchart that I have given, which actually can be the flowchart has the commands that can be inputted in Unix or Linux, and hence we can get the COBRA toolbox in our computer itself. There is another table here which shows the commands that are used. The highlighted commands are ones which are most important for GEM analysis. Program toolbox. That is something that can be seen on your terminal that shows you which where the manual is, what help buttons are, which flags to look out for, and hence it's very user friendly. At the bottom left corner, we can see there is a flowchart that shows how COBRA is actually utilized in equilibrium genome cell modeling. And here, as before, we have the important tool commands that can be used for GM modeling, as well as some other commands that can be used for just getting the flow of the program. For the prediction and analysis of metabolic pathways and genome scale networks. It predicts metabolic pathways and automatically reconstructs microbial metabolic models using a curated reaction database and a novel gap filling algorithm. The pathway prediction is based on multiple biochemistry database that comprises information on pathway structures, the pathway's key enzymes, and reaction stoichiometries. Moreover, GAPSEC constructs genome-scale metabolic models that enable FBA, that is flux-based analysis, metabolic phenotype prediction, as well as the application in simulations of community metabolism. We can install GAPSEC via two methods, that is in Ubuntu, that is a sudo apt install command that can be used, or we can use it in Conda itself, where there is multiple steps as given here. To test if the installation is completed, there is a single command set dot slash gapsec test. Shows the flow of the GAPSEC program. We give our genome sequence in FASTA format and it goes under pathway prediction, pathway analysis, transporter prediction, after which a draft network is reconstructed. From the draft network, there is gap filling stage where we can actually see where the gaps being filled, and then we get our genome scale metabolic model, which have our benchmarks and constraint based models can be based on these two. Similarly, as before, you can see there are commands in description. The first command, that is gapsec do all toy, actually predicts everything. But the problem is this takes multiple hours. That's at least 8 to 9 hours. Since our computer cannot handle this much time, we can use other programs, other commands as well, which so does each and every step separately and takes minimum time, and we can stop in between whenever we want. Operating system. The Unix OS was developed by Linux Torvalds in 1991, which further has an idea to improve the Unix OS. Previously, when Linux proposed the idea to Unix, it got rejected, and hence he made a completely different software by himself, and now we have our Linux. Uh, the first table shows the basic commands that are there in Linux that can be used to that everyone needs to know. The second table shows the essential commands like ping, unique. These are the commands which are based on genome metabolic modeling as well as other programs that you need to utilize. Linux has one good quality is that we can base our program and start from scratch, so it can be customized very easily. The references that I have taken for all the programs and learning GEM, and I hope you enjoyed the video. Thanks.